Hello dear students, welcome back to the biology laboratory. So we are discussing the third chapter, cell structure and its function. So before starting the laboratory practicals, let us know what is cell. So in your theory classes, through your audio clip, I have explained what is a cell. A cell is a single structural and functional unit of all the organisms. Means any of the living organisms, for the first, it starts from single cell. It's a basic unit. And based on that, we have unicellular organisms and multicellular organisms. What are unicellular organisms? Uni means single, cellular cells. If the organism is made up of single cell, then it is called unicellular organisms. Example, amoeba, bacteria, etc. What are multicellular organisms? The organisms which are made up of millions and billions of cells. They are known as multicellular organisms. As I told you earlier, none of the scientific terms are named simply. They have some specific meaning for that, right? So in today's class, let us study about the microscope. Prior to that, let us have a quick glimpse on what all we studied. It was Antony von Leeuwenhoek who first discovered simple microscope, okay? That they were using biconvex lens, we have studied in our theory class. Then later, Robert Hooke was a scientist who discovered the cell. It was almost like closed figure, isn't it? He found it in the cork. So Robert Hooke. Then he came to know that cell, and inside that there is a protoplasm. Protoplasm is a living substance which consists of cytoplasm and nucleus. And what is cytology? The branch of biology which deals with the study of cells. Clear? In today's biology practical class, let us know about the compound microscope. Okay? This is the compound microscope which we use to observe the unicellular organisms. That too, they are preserved in the slide form. Okay, these are all the slides. They are called slides. Okay. So this is a complete compound microscope which we use in our biology lab to observe the minute slides. See, these are all the permanent slides. They take the material and they just place it on the slide, and they'll be preserving for longer term. For example, you can observe unicellular organisms and some of the parts, dicot root, monocot roots, leaf, so many, right? Now, let us study the complete parts of compound microscope. Okay, this is the slide. Okay, first we'll come from starting. This is the eyepiece. Okay, this part is called complete eyepiece, where you just bend and focus, okay, to observe the slide which is placed here. This is eyepiece, and this is coarse adjustment. This is coarse adjustment, which is large. We can move up and down, okay, and this is fine adjustment, which is smaller one, it is fine, larger one, it is coarse adjustment, okay, and this is the slide table, okay, this is the slide, clear, and we have two clips here, we have two clips, it is movable, these are called slide clips. You can just place the slide here, okay, and slowly you can fix the slide. These are called slide clips, these are slide, and also we have the light source, the convex lens, right, where the light is you can receive maximum. It is movable in all the directions, okay. If possible, there will be some light operated compound microscope also. Okay. So this is natural light where you can focus and you can place this slide, and through this eyepiece you can adjust, that is through coarse adjustment and fine adjustment. Here we have two objective lenses, high objective lens, okay, the bigger one, it is higher objective lens, and the smaller one, it is lower objective lens. Higher we have 45 into 0.65, that is higher objective lens, we can magnify it, and here the smaller one, objective lens, okay, you can, where the material will be viewed in the smaller magnified way. Here larger, you can just see the internal structure also through this. Let me explain you once again. This is a compound microscope. We have eyepiece through which you can view the material placed here on the slide. Clear? And this is coarse adjustment. Okay. This is where you can handle it. Okay. This is fine adjustment. We have two objective lens, higher objective lens and lower objective eyepiece lens. This is the slide. Okay. Let me remove the slide here. This is the slide. It's a glass material, and also the cover slip. The material is placed and they have glued it. They have sticked it. Once you are going to place here, and these are the slide clips, you can just place it there. And this is the light source. Okay. The convex lens, you can just rotate it where the light source is comfortable for you, so that you can just focus on that particular material. So this is a complete parts of compound microscope. So dear students, in today's biology practical class, I have explained you about the different parts of compound microscope, which is used to observe the slides, the materials, right? Watch the video at least 3 times through your headphones and open your textbook and be very much perfect with the labelings of the compound microscope. So this is the starting class where you will come across this, and it will end up to your +2, that is 12th grade. So don't be very negligent about your studies. And shortly, in the month of March, that is from 20 or 21st, your annual examination is going to start. If you are not passing through that examination, definitely no promotions for you. Don't take the studies very lightly. Okay? Watch the video, children. Read the textbook. If you have any video, if you have any doubts, you can just call me. Take care, children.